Professor, please.
Okay, good morning, everyone. I'm Professor Hua Zhang from City University of Hong Kong. Welcome to a series of virtual academic seminars to celebrate the launch of SmartMath. SmartMath is an open access journal co-launched by Wiley and Tianjin University. I'm one of the co-editors-in-chief. The other one is Professor Wenping Hu from Tianjin University. Today, Professor Catherine Murphy is a speaker. Before her talk, let me briefly introduce her. Professor Catherine Murphy is a Larry Faulkner Endowed Chair in Chemistry at UIUC and the head of the Department of Chemistry. She earned 2 bachelor's degrees in chemistry and in biochemistry from UIUC in 1986 and a PhD in chemistry under the direction of Arthur Ellis in the University of Wisconsin-Madison in 1990. From 1990 to 1993, she was an NSF and then NIH postdoc fellow in the laboratory of Jacqueline Bolton at Caltech. Professor Kathleen Murphy joined the Department of Chemistry and Biochemistry at the University of South Carolina as assistant professor in 1993. She rose through the ranks at the South Carolina and then returned to UIUC in 2009 as a full professor of chemistry. Over the years, she has earned numerous awards. She was elected to the U.S. National Academy of Sciences in 2015 and to the American Academy of Arts and Sciences in 2019. Today, she's going to talk about a golden time for nanotechnology. Let's welcome Catherine to give a talk. Catherine, please.
Well, thank you very much. It's a pleasure to be here in this virtual world. And I want to thank Y and all the other organizers. So works okay. Okay, can everybody see that?
Yes.
All right, I'm going to go to full screen. That's what I had that problem last time. Let's see if this works. Okay. Yep.
Cool.
Okay, thank you very much. It's a yes.
Catherine, sorry,
yep.
We cannot see the full screen.
Okay,
happens.
But you can see this okay?
Yeah, this is okay.
All right, maybe I'll just make this really large. And then we'll try one more time.
Yeah.
Yes?
Yeah, good.
Okay.
Thank you.
Okay, great. All right. So let me tell you about what we've been doing in this area. So I want to start out by congratulating SmartMath for launching a new journal. It's very exciting to see. I look forward to, I guess some of the articles are already out, so I look forward to reading more from this journal, which is a great opportunity for those of us who want to publish more in active smart materials. It's very exciting. So today what I discuss is sort of look a little bit back and see nanotechnology, how it started, at least in the United States, how we got all excited about it, where people think it's going. And then I'm going to be focusing on gold nanomaterials. So I'll explain a little bit about why gold is so great for so many reasons. I'll go into the synthesis of the particles that we make all the time, which are gold nanorods, talk about some of the surface chemistry and some really neat experiments we've done recently to quantify ligands on the surface, the organic soft stuff on the surface of these particles. We have a bent toward the biological side of things, so I'll share some results we've had about nanoparticles at the cellular level and even a little bit of ecosystem work. And then in terms of smart materials, one of the main ways that you can interrogate and get these materials to do something is to shine light on them. So I have some results I'll share with you about some of the photothermal capabilities of these materials. So the National Nanotechnology Initiative in the United States started back in 2001. That's when there was a lot of funding, scientific funding for work in the area. And it's the NA still exists today, and they update every few years what the big directions are in the field of nanotechnology. When it first started, it was much more based on physics and engineering principles. But over time, what's really happened is the sort of the biological applications have really come to the fore. So if you go to the website for this initiative, and you find a PowerPoint presentation or brochure that they have, the very first thing they say is nanomedicine is one of the main promises of nanotechnology, right? That these very small nanomaterials will be able to do things that molecules can't do and things that larger objects can't do. So there's a lot of work in therapy, there's a lot of work in diagnostics, there's a lot of work in sensors, and then there's a lot of interesting work coming out in terms of environmental monitoring, environmental sensors, environmental cleanup, and things like clean drinking water is always a perennial problem in many parts of the world, and there are nanotechnology-enabled solutions. So, of course, as a chemist, little bits of metal, like the kind we make, have been used for decades in the field of catalysis. So that's a whole nother part of nano that's kind of been there, 50 years or so. But again, the bio side is where there's a lot of sort of new things emerging, which is very exciting. And one of those sort of phenomena that people have gotten very excited about in the last 20 years or so is drug delivery applications. And I don't do drug delivery per se myself, but we do a lot of things that are related to it. So I'll just give you the big picture here. The general idea is that a lot of drugs that people make now or in the recent past are small molecules that are somewhat hydrophobic, and therefore they're difficult to get to the target in the human body, which of course is an aqueous system. So nanoparticles can carry these insoluble drugs. It doesn't have to be metal like we make. There's liposomes, there's silica, there's magnetic particles, there's polymers, there's dendrimers, there's carbon nanomaterials, there's all kinds of things. But one of the big drivers here is cancer, which is one of the leading causes of death in the developed world. And so people for a long time have known that there's this enhanced permeability and retention effect. If you're trying to deliver things to tumors, where the idea is that there's leaky vasculature near the tumor. So objects that are 50 to 70 nanometers in size, or maybe 100, maybe 40, something like this, they will be more taken up by tumors. And so forth. They're just passively, without any chemical modification, there is some hope that the chemotherapeutic drug would actually get to the tumor more efficiently. And so that's been studied for now a very long time. And that's one of the original promises, of the field. And, of course, the biologists have studied nanoscale objects getting into cells or into tumors and so forth for a long time, although the nanoscale objects were usually viruses or maybe bacteria on the micron scale. And so there's a lot of knowledge already in the literature about what size object would be getting into cells through different mechanisms. And so there's generally, we would maybe just say endocytosis for objects getting into a living cell. But there's lots of different kinds of endocytosis, and I have just some of them listed here, and they have different names. But they're kind of a function of the size of the object. And how the cell processes these things and eliminates them is actually a whole other subject of work, which is really pretty interesting as well. So in vivo, if we think about the human body, and you wonder how small do you have to be if you took a capsule that had particles in it, how small would those particles have to be to clear all the way out of your system with, and not linger. People would say those objects should be less than 10 nanometers in size, which is a little bit of a difficulty since I just told you that things that are 50 to 70 nanometers are actually good in terms of tumor, potentially uptake. So now over time, people have developed a lot of strategies, right, for things that dynamically evolve in time that might be one size at one stage, and then as they go through an environmental or biological processing, they can change their overall size. So that's something of a little bit of an introduction to, the scope that nanotechnology has on the biological side, which is a huge scope. So in my lab, we focus on gold. And I want to just point out that artists were way before the scientists here when it came to understanding that finely divided matter, different properties than the bulk. So here's a picture of me at the Institute of Chicago, and I'm looking at this cup here. So this cup has got metal work on it, and then it's got glass in the middle, and the glass is not clear. It's this kind of rosy red color, and that's due to colloidal metal particles that were dispersed in the glass. So this cup is from the year 400, roughly, and so this is a Roman artifact. What the Romans did is they would take gold, the element, which you can isolate, beat it with a hammer till it's very thin, and disperse it into molten glass that they would pour into this cup. and it would disperse to give this reddish color. And so right away, the Romans knew in the year 400 that finely divided gold had different optical properties than the bulk, which they didn't call it that. That's what they were observing. So the Romans called this pigment red gold, and later on you find it in stained glass windows and stuff too. So, organic vegetable dyes are not going to survive hundreds of years in sunlight, to maintain their color. So the metals, of course, are very good here. And I'm an inorganic chemist by training, so naturally I'm very happy that the metals do this. So we can fast forward to Michael Faraday, scientists in the Victorian times, and his chemical bench is in this museum in London. And so these are his solutions from 1857, sitting here on this bench. And he's the first person we know of who made colloidal gold chemically. So he does what we end up doing now. He takes a metal salt and reducing agent in solution to make these finely divided particles that are suspended and have been suspended in this solution since 1857. So in his case, the reducing agent was elemental phosphorus of the gold salt, and his solvent was carbon disulfide. So anyway, so those things are sitting there. And again, we have this reddish color. And so at this point in the game, we have a pretty good understanding of why finely divided metals would have properties. It goes back to several different things. You can think about the mean-free path of an electron in a metal. If you look up a standard solid-state physics textbook, that would tell you that the numbers here are like 10 to 100 nanometers. So that gives you a length scale roughly where things would be interesting, right? If you shrink your grain size of your metal down below a few hundred nanometers, things would be interesting. You can also do what Gustav Mie did over 100 years ago and solve Maxwell's equations for a certain case, the small conducting sphere. So just like we have in chemistry, right, we solve Schrodinger for the particle in the box, a simple case. Without a computer, you can solve Maxwell's equations, which tell us light is electromagnetic wave. For the case of the small conducting sphere, and small here means smaller than the wavelengths of visible light, so hundreds of nanometers, conducting meaning metal, and sphere meaning sphere. And so here's a picture everybody shows of what we now call the plasmon. So here's my electric field vector of light. And when I'm in tune with the resonant frequency, these loosely bound electrons that you need to be a metal or conductor, you get this huge oscillation of these electrons in a coherent manner over the surface of your particle. So people call this the plasmon now, and that's responsible for the brilliant colors. So in our case, we never want to be too small and we never want to be too big. If we're too small, we don't have enough electrons to support this phenomenon. So then you lose the plasmon. So below 3 nanometers or so, your gold nanoparticles do not look reddish anymore. And if you're too big, the particles just look like a big hunk of gold. So between the 4 nanometers and 300 nanometers, something like that, that's where we're going to be because we want the plasmons. And so one of the things that we're known for in the group is making these gold nanorods. And I'll explain to you how we do that. This is a pretty old slide, and as I'll show you, we can make, we keep making better particles all the time. So, and the synthesis is much like Michael Faraday. We take a metal salt and reduce it to the metal, but we do in a controlled manner to grow materials in isotropic shapes. And I'll talk a little bit about that too. So here's a solution that's about 1 nanomolar in particle concentration, and these are almost spheres. And you see here's the same reddish color that we had in the Michael Faraday solution. And if you take a spectrum of these things, you find a nice peak for your plasmon around 520 nanometers, which if you calculate the thing like Gustav Mie does, that's totally spot on for, in this case, 20 nanometer spheres of gold and water. So you plug in the numbers for all that stuff and you're good to go. So what we've been able to do over time is develop methods to make elongated particles, and I'll show you how that works in a little bit. And when you do that, you get this rainbow of colors that corresponds to the shape of the material on the nanoscale. And if you take spectra of these things, what we find is that our one plasmon band has split into 2. And so how people understand this is we're absorbing and scattering light along the short axis of the particles, which is pretty much the same as we go through the series here. And that's the band that's about 520 nanometers. And that's the so-called transverse plasmon band. And as we make the particles more and more elongated, now the washing along the long axis of the material, and that's the longitudinal plasmon band. And that goes with the overall shape of the particles, so that peak goes to longer and longer wavelengths or lower and lower energy the more elongated the particle is. So unlike quantum dots, where the size of the dot really determines the optical properties, here it's much more the shape that does that. So these solutions are all about 1 nanomolar concentration. So these are really good absorbers compared to most molecules. Down here, I'm showing you this little picture that looks like a galaxy. But what it actually is, it's a darkfield optical micrograph of our nanorods immobilized in a gel. And so we're just sending white light into this little body, and we're just looking at the light that's just scattered out to the side. So it's just Rayleigh elastic light scattering, why is the sky blue kind of scattering, and all the little stars in the sky are the particles here, and the misty colors are from the gel because it's kind of turbid. So the field of view here is about 3 millimeters, so you can't literally say that one spot is one particle, but I'll show you later. We've done plenty of imaging experiments where, in fact, you can discern what appears to be, by Browning in motion, a single particle, even in an optical microscope kind of experiment. So, so in my lab, some students are very interested in just how the particles are made, wanting to understand mechanistically the inorganic chemistry and shape control and crystallographic axes, and we've done a fair amount of work in that area. But many other students want to make their particles and get on with it and do some applications. And so here's where it becomes very interesting to think about what happens when you shine light on these materials. So here's my little gold nanorod, and I'm shining light. Okay, so I've got these plasmons. Okay, depending on what wavelength of light I shine, I'll be exciting the transverse or longitudinal modes. And what this does for you, it does a number of things. So while you're shining light, you're moving all these electrons around, so that does create a local electric field. And any molecule that experiences a local electric field will have its optical properties altered. So the most famous example of this is surface-enhanced Raman scattering. So Raman, of course, is a vibrational technique, very low cross-section. But if you have your molecule experience a big electric field, the Raman cross-section goes to the 4th power. So now you can start seeing things you couldn't see before because the molecule is experiencing an electric field. So this is an old paper we have, but I'll just briefly show you here. So here we have a flat gold film with a self-assembled monolayer. And if you try to do get Raman data, those molecules in a monolayer and the spot size of your laser on this flat surface, there's no hope, okay? You get no signal. That's the top one here. But if you sprinkle some particles on, which are much bigger than the molecules, by the way, you sprinkle some particles on, and now when you shine light, the Raman laser here is 785 nanometers where the particles absorb, and so now you start seeing all the ring-breathing modes of the molecules light up for different sorts of particles that you put on, because you're creating junctions between the particles and that flat metal surface, and that really amplifies the effect of this surface-enhanced Raman scattering. So that's a well-known phenomenon. It's always been difficult to be quantitative about that. But nonetheless, the sort of the limit here, right, would be sort of single molecule detection. And because you're looking at vibrational features, you have really great diagnostic capability. So up here, we're making a bad fluorophore good. So there is a phenomenon also of surface-enhanced fluorescence. And here what we're trying to do is put the molecule at the right distance from the surface. Because if you put a fluorophore too close to the metal, it'll just be quenched. If you put it too far, it won't feel the electric field that you generate when you shine light on the plasmons. So in this case, it's hard to see here, but we put little glass shells, silica SiO2, silicon dioxide shells, and we could covalently attach our dye in this case and define distances from the particle surface, and then show, in this case, that you get about a 10-fold boost in the emission of your fluorophore if you're the right distance away from the metal. So anyway, the electric fields, that you generate can be used for sort of diagnostic type capabilities. But another thing that happens all at the same time when you're doing these shining light experiments is you generate heat. I mentioned how the particles are sort of 1 nanomolar in concentration. They're very brightly colored, so they're very strong chromophores, but they don't fluoresce themselves, really. So what will happen is they'll absorb all this energy and then dump it heat to the local environment. And you can use that heat, and I'll mention this at the end of the talk, to do things like pop molecules off the surface that you put on there. And again, we don't get to the animal level, but again, there's a lot of excitement about therapeutics here, where if you could get your particle to, say, a tumor and shine light, then you can ablate the tumor, and no tumor is going to be chemoresistant to heat. So there's a lot of excitement about this very early on, I would say, and then it kind of died down, but now it's starting to come back up again because now there's human clinical trials that are showing efficacy of doing this exact thing in prostate cancer situations in human patients. So that's very exciting. So all the excitement here has not gone unnoticed by commercial companies, and so now there's a lot of little startup companies that make gold nanorods. Okay, so you don't have to ask me for them. You can buy all these different places. This is the place that's using our synthesis here, nanoparts. But if you look at how they advertise their uses of these particles, it's for imaging. That could be due to the scattering, like that little galaxy picture I showed you, or could be doing some of this Raman stuff too. Or, medical treatment, that's the photothermal therapy. Here's hyperthermia, that's the photothermal therapy. And I do want to mention that, at least in the US, I don't know about China, colloidal gold solutions are sold as nutritional supplements, which is not true. There's no nutrition here, but people do buy them and people do drink them. So I keep asking any colleagues who work at medical schools to, find these people, find people who drink this stuff. Let's do it. Experiment to see how they are different from people who don't drink this stuff. I'm guessing their livers are probably full of these 20 nanometer colloidal gold particles. In terms of practical advantages of using gold as your nanoparticle for things, I want to also point out that you're doing environmental, you have very complex systems. It's easy to find the gold. It's got a large, of course, atomic number, so if you do a lot of electron microscopy, it's easy to find it. But also, if you want to digest samples, ICP mass spectrometry has a part per trillion detection limit for gold. So for our size rods, for instance, that corresponds to like one dozen rods per liter can be detected if you digested all the gold there. That's a really low detection limit. So that makes quantification pretty good. Compared to the other metals in the periodic table, right, gold's very stable and noble, oxidized under most conditions. And of course, nowadays, right, over the last 20 years, people have developed many ways to make different sizes, different shapes. So we do rods, but there's prisms, there's stars, there's platelets, there's all kinds of things that people have made. And the surface chemistry possibilities are also pretty amazing. So, okay, so how do we make these particles? So we do what Michael Faraday did. We take a metal salt and reduce it to the metal. But we're going to do it in 2 steps in order to control the crystal growth. So I start out here with my metal salt in water, in air at room temperature with this surfactant here. It turns out the surfactant here is not really necessary at this stage, but this particular prep that I'm going to show you the data for, yes, it was there. And then what we do is we add a strong reducing agent under basic conditions that's a little bit chilly. And then we go ahead and quantitatively convert all these gold-3 ions into gold-0 atoms. In the solution now, which is metastable, so it'll last, maybe 1 to 5 hours, something like this. And it has this brownish color because these little particles here are too small to show the plasmons. Okay, so they're tiny little seeds is what we call them. Okay, so we do that step, and then meanwhile, we make here of the same metal salt. Now with the surfactant is important, so there it is. And now what's interesting here about the inorganic chemistry side of things, we have an active metal ion here, much lower concentration than the gold that we're going to use here. In this case, it's silver nitrate. And what we've learned over time, and you can ask me this later, a little bit of silver operationally will give you a short rod, and then more silver gives you a longer rod. So this impurity controls the aspect ratio of the final product. It's pretty interesting, right? So anyway, we put in our metal salts here with the surfactant. We add now a weak reducing agent. So in this case, it's hydroquinone. Hydroquinone's redox potential is insufficient to reduce gold 3 to gold 0. But the idea is when we add the seeds, now the reduction potential of the adsorbed metal species is different. So now you can actually grow more, you can deposit on the pre-existing seed particles. So you don't want to make new seeds in this step. You don't want to use a strong reducing agent here. And you don't want to have a thiol, because a thiol is a strong binder to gold, and you're just going to make spheres. So if you want it, so it's a delicate balance. You want to have a ligand that's going to bound to the sphere weak enough so that you can grow, but not so weakly that it just desorbs all the time. So anyway, when we do these experiments with those exact conditions I just showed you, using hydroquinone as our reducing agent, normally we do ascorbic acid, you can push the plasmons out pretty far, 1200 nanometers longitudinal mode, and that's getting close to 1500 nanometers telecommunications wavelengths. And the particles are just gorgeous. We just never get tired of making them because they're just beautiful when we look at them by electron microscopy. Now, when we're done, we got these suspended particles, and for most of the, for early on, all we cared about was the plasmons and the metal, but then, of course, there's all this other stuff on the surface, right? So what is it and how much is there? It's actually kind of a question which becomes important, especially for biological applications, because the surfactant on its own is toxic to a lot of different cells at sort of 200 nanomolar concentrations. So anyway, here's our particle, and we've done a lot of experiments over time, okay, to look at what the surface is like. And so here's our picture right now. So here's our metal surface, and I have a little bit of silver in these particles. You can quantify that if you digest them. So yeah, there's a little bit of silver in them. And we have chemisorbed bromide first at the surface, and then we have ion pairing interactions with our reactants. We have a ton of it when we do these reactions. And then we have then because we're in water, these hydrophobic chains want to be away from water. You end up building up this bilayer, at least a bilayer structure. Or Jason Hafner at the university would say it's a micelle, an actual ball of surfactant. And that is possible. It's kind of hard to tell from some of these data. So anyway, but we have about a 3 nanometer hydrophobic region, okay, near this hunk of metal with a quaternary ammonium head group, okay, facing the solvent. So it's actually much more complicated than you might think. And once you realize this stuff is there, you can think about putting molecules in there, trying to cover this up, or try to do substitution reaction to displace this to put on other things that you care about. So one thing that we're very excited about recently is we have this cartoon here you might see, right, where it looks like I'm implying that there's more ligands on the sides of the rods compared to the ends. The reason for that is if you try to add do some assembly of these particles, a lot of experiments, including some by us, seem to indicate that the ends of the rods are easier to substitute with the thiol compared to this surfactant stuff. Why would that be? Well, one reason would be, maybe there's fewer molecules on there, and so there's less van der Waals forces holding the chains together, and so therefore they're more easily displaced. Okay, that's a hypothesis. So we wanted to test. That's a hypothesis we've had for a long time, but we actually have only recently do we have sort of more evidence for it. So here's an experiment we did recently with Pinsheng Huang, my colleague in materials science, and her student Blanca. So what we're going to do here is we're going to deposit these particles, which are only stable in water, on a single layer graphene grid, and we're going to do a very high quality electron microscopy here in a STEM EELS experiment. EELS here is energy electron loss spectroscopy. It's a way to image light elements in TEM. So what we're going to do is calibrate EELS signal for carbon on the single layer graphene grid, and then we're going to move to a particle and go all the way around and quantify how many carbons per square nanometer we have for 500 particles, and see if it's true that we have fewer organic molecules on the ends of the rods compared to the sides. Okay, first problem is water will destroy single layer graphene grids due to the surface tension, and our particles have to be in water. So a student that I had recently in the group, Josh Hmmm, has a twin brother named Jordan. And Jordan was in Ken Suslick's lab, and Ken Suslick has done a lot of ultrasonic spray pyrolysis in my department. And so the two of them got together and built this little setup here where we have a nebulizer, which is like what you would find in a water humidifier. And this will produce tiny droplets of water. And so what we do is we put our solutions of particles in here, and we have a carrier gas, and then we blow it through this tube, and then we put our TEM grid that has the single layer graphene grids on it over here. And the idea is that we will gently waft on a very small drop of water that will not destroy the single layer graphene grid, and therefore we can do the experiment we really want to do. So in fact, that ends up working after much trial and error. So in sample A here, these black holes is where a single layer graphene grid was, and we just put a drop of water on like usual, and then it destroys them all. But here we do this little nebulizer thing down here in part B, and all the gray you see here, those are single layer graphene grids that survived the aqueous system. And so then you can zoom in and zoom in and zoom in, and now you can see the metal. You can see that there's some organic stuff on here. You can look at the graphene easily by EELS and so forth. So when we do these experiments, it's very exciting. We find So here's an example of a single particle image. So the bright spots here are how many carbon atoms per square nanometer we have, going all the way around. It's pretty exciting to be able to do that, right? And at the max, the density of these ligands is about 9 molecules per square nanometer, which is pretty much the biggest density of organic matter anyone's ever seen on gold. And you get about a 30% drop of the rod. There's actually a lot more than I thought it would be. And it's actually not quite clear why this is the case at this point, but it is the case that, in fact, there is less stuff at the ends of the sides. So anyway, this is turning into a whole other thing in the lab in terms of quantifying how much stuff you have at different points, leading to some very interesting chemistry work based on the anisotropy of the particle. So we keep exploring these rods, and on the biological side of things, the absolute size of your object makes a big difference. So the particles I just showed you were maybe 10 to 15 nanometers in diameter, and then the lengths are tunable, 20, 30, 40, 50, 60 nanometers, something like this. That's kind of what we're doing up here, our normal method, something like that. My student Wei Chang, a couple years ago now, developed a synthetic method to make what we call mini rods. So what's the difference here? You add way more seeds in the growth step. Okay, that makes sense. For all the concentrations being the same, the particles will have to be smaller, right? But the other thing she found you had to do at the front here was add some HCl. And you might think, oh, we're just adding acid. But again, with all these complex equilibrium solution, it's actually more complicated than that. So ascorbic acid is our weak reducing agent here. Its redox potential depends on pH, so HCl is going to affect that, yes. But now we're adding a lot of chloride. And if you look at the speciation here of our metal, we started out with gold chloride. We add a lot of bromide. Okay, and these ligands are exchangeable on the gold 3 ion surface, or, ion. So in fact, what you're doing is you're shifting the equilibria of your gold halide molecular complex is more to the chloride side, and those redox potentials are all different from the pure bromide and the pure chloride. So anyway, the equilibria here are pretty complex. Nonetheless, we were able to make these mini rods in really high yield and really small sizes. So now the diameter is like 8 nanometers or 6 nanometers, and the lengths are maybe 20 nanometers. And like I mentioned earlier, the absolute size does make a difference if you want to think about clearing, out of a biological system. The other thing that these mini rods buy for you is if you want to do the photothermal heating thing, they're much more efficient at doing so. So the bigger a particle is, the more it'll scatter light. So that's kind of shown over here from this other group here. That they've done a calculation of what the regular rods, regular size rods do. So the extinction is the sum of absorption and scattering. That's what you would measure in your spectrometer. And then you can calculate or do somewhat more painful experiments to get the scattering and absorption components separated. And for a sort of a normal sized rod, half the photons are just scattered and half are absorbed to then produce heat. But for these mini rods, almost all the photons are absorbed to then produce heat. Although if you look at the scales here, you wonder, the short rods, still the what they call short rods over here, the absolute magnitudes are quite different. So anyway, that's something we're working on too, is being more quantitative about this. Anyway, so there's that aspect. So if you want to do imaging of the particles by light scattering, you want a big particle. If you want to do photothermal heating, the general idea is you want a small particle. If we look at things like cellular uptake, again, this is from another labs group here. They've got big rods and small rods with a protein coating. And so, yes, more small rods get in, and then they clear fast into a big rod of the same overall shape. So the surfactant we have on the surface is really interesting, right? I just showed you some cool data. But for biological applications, it's kind of a problem because if it's going to desorb, right, for sure, there's going to be some equilibria there. And then that molecule by itself is going to be problematic for a lot of biological systems. So we have learned over time there's lots of ways to deal with that. So one way is just to cover it up with polyelectrolytes. So this is stuff from the late 90s that people developed back then, C. Stecker, other people. Paula Hammond does this a lot on her surfaces for biological implants. So it's a layer by layer polyelectrolyte, in our case, wrapping around colloidal particles. So there are these polymers you can buy commercially, polystyrene sulfonate, polyacrylic acid, and then these amines here. And so you can go ahead and incubate your particles and then build up these layers. And you can tell by the zeta potential, effective surface charge, that you have cationic, anionic, cationic, anionic, cationic, anionic, as you build up these layers. One problem here is, compared to a flat surface, it's hard to separate the polymer from the particle when they're both around the same size, which is usually the case here. So usually we go up maybe 10 layers, and then we sort of start losing a lot of sample. But then you have a lot of opportunities to do chemistry on top of your polyelectrolyte layers. So you can terminate in acid groups or amine groups and all kinds of things. You could use peptides. That's kind of expensive, but you could use peptides as your wrapping layer as well. So we've shown that before too. And those are all examples of soft shells, we could say, right? Organic surface groups. It's also possible, like I showed you at the beginning when we were talking about fluorescence enhancement, surface enhanced fluorescence effects. Here's a zoom in of some of these little silica shells. So again, we've got some very nice methods to grow pretty well-defined silica shells here. And these are mesoporous. You can almost see that in the micrographs here. And so this is another way to mitigate the problems you might have with your surfactant. In this case, mesoporous silica uses this exact surfactant CTAB to make the pores. So it's kind of a one good thing about that surfactant. You can use it to template the formation of another material on top. If you want to just straight up replace this surfactant, again, Japan in particular, that it's hard to do with just a simple thiol straight up. It's easier to go through a large water soluble polymeric thiol for reasons I don't quite understand. But anyway, indeed, you can go through and add a PEG thiol here and then come in with your thiol of interest to replace it on the surface. And this PEG stuff becomes very interesting when we think about trying to do more interesting chemistry on the surface here. So I just showed you we can grow silica all the way around the particles. And I won't go into detail here, but I will say that we've developed some methods, riffing off of Jianfang Wang, who's at Hong Kong too, about how to grow materials just at the ends or just on the sides of these gold nanorods. So here we have silica just depositing on the ends or just on the sides. And this is another EELS image here where you can see the silica looking purple and carbon, that surfactant, is in the pores looking green. So you can see that these pores on the longitudinal side are almost radial, right over here, kind of radial pores. And then if you think about molecules getting into the pores, which they could, then we again have opportunities for sensing and diagnostics and so forth. So if we think about the bio side now, think about biomolecules and cells, what do we expect to have happen? Well, these particles are about the size of a virus, right, or an organelle. And so there's kind of a scale drawing of what it might look like. And there's a lot of things we can predict, right? It's kind of, you can see these little particles are irritants to the cell. So we'd expect, right, some inflammation mechanisms will be turned on, clearance mechanisms will be turned on. One thing that we're very interested in doing, exploring, we've got some interesting data on, although I don't have time to show it all here, is that since these particles have large surface areas, any molecule that binds to them is now less bioavailable to the cell. And so that is another way that a nanoparticle can influence the cell, right? It's not that the particle is active. It's just that since it's taking something away, it's like a molecular knockout as far as the cell is concerned. So if you make that specific, that's actually pretty interesting. The other thing that, of course, for these particles for us, right, they're hard materials compared to the soft tissue of the cell. And again, there's a lot of work on phenomenon of mechanotransduction, how cells respond to the mechanical nature of their environment. And so again, we've done a little bit of work in that area. And our particles, by and large, don't dissolve, right? But other particles do dissolve in aqueous solutions, and that means they can release things, including their own core, right, of metal ions, perhaps. So again, there's lots of different mechanisms you can imagine what would happen when you put a particle inside a cell. But at this point, people also know, right, that if you just take your particle and put it in a biological fluid, before it even gets to the cell, there's going to be absorption of many components to the surface. So people call this now the protein corona. And again, now it's been 15 years or so since some of the first papers in this area. So certainly on flat surfaces, people knew if you put a flat stent, in a biological fluid, proteins would absorb, and you could quantify that by ellipsometry and so forth back in the 60s. But doing those same experiments on a colloidal particle surface was much more difficult. So again, people kind of rediscovered some of these phenomena for the particle world in around the year 2000 or so, and now there's been many reviews of such things. But just as an example, let me just point out this paper of ours from quite a while ago here. So here I've got these little rods. So cationic, anionic, cationic. I did my little wrapping thing. And then if I put them in a solution that contains serum proteins, the zeta potential goes to the same place in about 5 minutes. And this zeta potential here corresponds to the zeta potential of the most abundant protein in the solution. So even if you're imagining that you'll be able to target something because your particle has a net charge, sure, it has a net charge, but now it's buried under all this other stuff. So this does mean that when we try to connect the dots at the molecular level, it's a little more complicated than we might think. We know what we put in, and we maybe can measure some response, but knowing how we got response is actually a pretty interesting journey to make. So what I want to show you here is one of the uses of these particles as imaging agents. So this is a darkfield optical microscopy. So what we're going to see in this little movie I'm going to show you, we've got cells in a little dish, and the dish is full of collagen, which is a soft protein, and the cells are adherent, so they're crawling around, they're doing their thing. And what we have in the little dish is a lot of particles all over the place on the surface of the protein surface, all around in the fluid, and we're just going to watch the cells do their thing as they crawl around. And the cells are not staying in this movie, so they're hard to see, but what's going to happen is that they suck up these particles and get brighter and brighter and brighter, and you'll see dark trails left behind where the particles were. Okay, so let's see if this movie works. So this is sped up by about 8 from real time. And so this is darkfield optical microscopy. Again, a very easy kind of experiment to do. And one problem with darkfield is anything that scatters light shows up like that little hunk, this little hunk of salt or whatever it is floating around. But you can see that these cells are now much easier to see. You see, it looks like they have a big hole in them, right? That's the nucleus, which is pretty big. And, I don't know, if I was a cell, I wouldn't want to eat this heavy, but I eat what they're doing. So you can do these experiments for lots of different kinds of particles, different kinds of cells, and then you can learn something, right? So what did we learn in this experiment? So here's a set of cells. These are PC3 cells, prostate cancer cells. You can buy them. It's a vanilla flavored cancer cell line. And black, and we're looking here at mean square displacement over time. So black is no particles. And then if you feed these cells any particle, it slows them down, which makes sense because they're taking up all this extra mass. You can scrape off the cells and quantify by ICP-MS how much gold you've taken up, and it's like 500,000 particles per cell. It's a lot, which is more than I thought these things would be doing. But then you go to these cells, HDF, human dermal fibroblasts, very boring skin. Okay, human skin cells, nothing fancy there at all. What's kind of weird is that black is no particle. So some particles make the cells slow down, and some make them speed up, okay, depending on the surface chemistry. Okay, so this is an example of one of these things where you know what you put in, and now the question is, what is responsible for it? So what we're doing, the cells are not dying, okay, they're just changing how they move. And if you're changing the behavior of a cell, that means you got to dig into the sort of genetic expression thing going on. So we do this with some microarray mRNA work here and so forth. So I've got here a whole bunch of different sorts of particles. Some of them have lipids on them, so they look more like lipid vesicles. And then we get data like this, which I'm not going to describe very much in detail at all. But we're looking at the expression of 14,000 genes here. And red is upregulated. Blue is downregulated, and we got the same kinds of particles down here, and for each of them. And so there's only 2 points to make here. Number one, lots of stuff is changing, but it's not changing enough to kill anything. And then number two, going from one cell type to another, very hard to predict, right, what's going to be changing. So what this has started to make us think about is, if you think about things like asbestos, right, that's probably the most common example of a toxic material, right, is asbestos. It's micron scale needles of a very certain composition that bury themselves in the lungs of people who breathe them. And asbestos doesn't kill you right away. It takes a long time. So we were wondering about these little particles and seeing these weird effects on these cells. What would happen if you expose these cells to particles for a very long time? I mean, would they adapt, or would they, get sicker or something like this? So anyway, in the experiment here, we took these same HDF cells that kind of behaved weirdly in terms of their migration ability, and we incubated them with the most innocuous particles we have here, the most boring particles, right, that shouldn't do anything, right? Gold is not going to dissolve, right, we would say. And we have anionic surface coatings that seems to be much more biocompatible than cationic ones, for instance. So we do our layering reactions on the rods and so forth. So what we do in the experiment here is we expose our cells for 24 hours to these particles at a very low dose. Then we wash the particles away, and then we let the cells go, not for 24 hours, not for 48 hours like usual, but we let them go for 5 months, which is a long time to do a cell experiment. Or we take the same batch of cells and expose them to these different particles here for the entire 5 months at a very, very low dose. So instead of changing out the media with fresh media, we keep putting particles over and over and over again, the same low dose for a chronic kind of exposure. Or we just have the media for the cells. And so what we can do, of course, is quantify uptake after the 5 months here, which is a long time, 20 weeks. And what you can find again is that, again, 35 times 10 to the fourth particles. So anyway, but what we're saying here is that the composition of a microbiome might be altered by the presence of nanomaterials. I think that's a very interesting starting point, right, for a lot of new work coming along. Now, I'm a chemist, right? So, to me, a lot of this very, advanced omics type data, what that says to me is that, okay, this means we can have testable hypotheses at the molecular level, right? We can look at the genes, what's their function supposed to be, like unfolded protein response, and then think about things like this. Okay, here's my folded protein, and then I have some intermediate, and maybe by being on a particle, I'm stabilizing a more unfolded looking conformation, and that is having an effect on the cellular function. Right? So what this means is we can try to go back to the molecular level and think about how we can control display of molecules on our particle surfaces. So this is the conclusion of a study that we did by NMR, 15 labeled protein. The protein here is alpha-synuclein. It's a very interesting protein, one of the most abundant proteins in the human brain. And it has no known function, but it does appear as aggregates in the brains of people with Parkinson's disease. So this protein, when it aggregates, that's thought to be the cytotoxic agent in this neurodegenerative disease, which is, again, a long chronic disease. It's not an instantaneous thing. So anyway, what we've done here in collaboration with my colleague Chad Rienstra, who's a solid-state NMR spectroscopist, so he expresses this protein, N15 labeled protein, and we can incubate it with our particles. And what we found is that when you use a citrate as the very boring, again, anionic little molecule here as a surface coating on just spheres now, the C terminus of the protein extends into solution, and it's an unstructured protein, and the C terminus looks just like the regular protein, but you can't find the N terminus because all the residues in the NMR are broadened, so you can't see them anymore. The N terminus is facing down. We've also done mass spectrometry experiments to chew the protein, and you find the same result. But it's interesting, if you have a quaternary ammonium surface on your particle, now you flip the orientation of this protein, and now the terminus is facing the solvent, and the C terminus is facing down. Where the aggregation happens in this protein is in the middle, okay, between the interfaces there. And so more recently, my student Sophie McLean in the group has been working with Chad Reinster's group to look at mutants of this protein, which are known to have familial Parkinson's disease phenotype in humans. And what we're finding is that you can compare type protein to how it sits on, now we're making like lipid-looking surfaces here, because one thing people do know is that when this protein binds to lipid vesicles, it goes alpha helical. And the question is, does it go alpha helical on a lipid-coated hard surface like this? And the answer seems to be no. But you can also figure out how it's oriented on the surface and what residues are more accessible to the outside world. And what's interesting here is that these residues and these mutants, these are the ones that are thought to be involved with the protein's ability to aggregate. So what this could mean, right, is that if you're trying to do some minutes and on a solution of this. And if you have particles and light together, so this is a little live dead cell staining kit, so green is alive and red is dead. And so if you have particles alone by themselves, they don't kill anything. Light alone doesn't kill anything, but particles and light together, yes, you kill the cells. And bacteria have cell walls, right, in addition to membranes. And you've blown big holes in their cell walls by doing this experiment. So bacteria, of course, can be in their planktonic form and swim around. That's what we did here. But a much harder problem is if they're in the biofilm form, right? And they've colonized a surface and they're more resistant to a lot of antibiotics and so forth in that way. So we've done a little bit with trying to see if our particles can break up biofilms, but that's a pretty tough problem. On the molecular side, though, a much more common thing to do is think about, releasing molecules, right, with this photothermal heat. And what's interesting now, I mentioned most drugs are small molecules, hydrophobic small molecules. That's why a nanocarrier is frequently useful. Turns out in the last 20 years, people are going more and more on the pharmaceutical into biologics, meaning a protein, right, frequently. And if you look at where we are here with the patent literature, 2020 is a year that's going to be memorable for many reasons. But one reason is a lot of these protein or biologic-based drug formulations are coming off the market. And so what this means is there's going to be a lot more activity for people trying to figure out how to deliver these things to the target of interest. So if we think about shining light and